Thank you so much. I want to really thank the Staglin family and One Mind, and for all of you for coming here to listen to the scientists. We love to be able to share our research with you and get to hear your questions and speak with you. So I'm looking forward to a really exciting afternoon. So I'm a developmental neurobiologist. I've long been interested in understanding how the brain wires up during development, how neural circuits are sculpted by experience, and how this wiring process can go awry in neurodevelopmental and neuropsychiatric disorders. Now, it's challenging because we, in people, can't just go in there and ask when things go awry, which circuits are vulnerable. You know, this has been a challenge in general. So what's really exciting, I think, right now for the field is emerging genetics. Human genetic studies are now identifying pathways and genes and loci that are giving us clues about the biology, and even more so, how we might harness this information to try to come up with new therapies and new biomarkers. Now, it turns out that some of the genetics more recently has been implicating a pathway and a cell, the cell that you see here called microglia, in some of these disorders, including schizophrenia and Alzheimer's disease. And what I want to do today is tell you about some of the work we've been doing on studying this process called synaptic pruning, and a group of molecules implicated in the immune system, and how understanding how this works normally is providing new insight into disease. Now, as you probably know, development is an incredibly plastic time. Anyone that has children knows children's brains change dramatically from the time they're a young child. And this plasticity is why my daughter can learn French seamlessly and why I cannot, right? And it's really remarkable. And this plasticity continues actually on through adolescence and into early adulthood. Not all adult development is happening at the same time in the brain. There's actually windows or critical periods, and it's also happening in different parts of the brain. And this is illustrated here in this human brain imaging study that was carried out by NIMH investigators and been shown both in animal studies and in human brain that this brain development is happening over periods of time that suggests different regions of the brain are developing at different times. And this is showing here in different colors where you can see that the maturity of the brain, they are actually measuring gray matter volume, that is illustrating, it's an index of maturity. And it turns out, if you look at this image here, if you compare in the same individuals they were tracking longitudinally over time, what's clear is that a 5-year-old brain, if you look at an area like the prefrontal cortex, it's continuing to mature into early adolescence. And so this is an important point because parts of your brain that are important into vision and sensation, they develop relatively early in development, but the parts of your brain that are involved in executive dysfunction, decision-making, all of these things are continuing to happen into this adolescent period. And so that raises the question of whether this plasticity, while important for learning, might also be a reason why there's vulnerability. And you just heard this talk by Erin, and that this vulnerability and this window may be related here. So the question we were asking is what are the mechanisms regulating this process? When you're measuring gray matter volume changes, what is in gray matter? What are we talking about here? Well, it turns out that this work, this idea of changing in the brain, has been long recognized even through anatomical studies. And the idea is that we start off with an excess of synaptic connections, the connections between neurons, and we start out with more connections early on. And through a normal developmental process called pruning, a large number of these extra connections get permanently pruned away, while others get strengthened and maintained. It's this idea of use it or lose it. Connections between neurons that are meaningful get strengthened, and connections that are less meaningful get pruned. And this pruning process is a normal process that allows us to have precise brain wiring. And defects in this pruning are thought to underlie disorders such as schizophrenia and neurodevelopmental disorders. So the question is, what is regulating this pruning process? And so it turns out that every neuron, of course, makes lots of synaptic connections. And it turns out that synaptic loss, or the loss between neurons, is a hallmark of many other CNS disorders, including schizophrenia, as well as Alzheimer's. And so the loss of synapses is one thing that we may be making the brain vulnerable. And so there was a number of imaging and other studies that have suggested the possibility that too much pruning during these critical periods could be underlying the pathobiology of some of these disorders. So the question is, what are the mechanisms? And the question that we've really been focused on is a really fundamental one, which is, how is it that synapses normally get eliminated? And more specifically, which synapses get eliminated? Now, of course, the synapses, or the connections between neurons, are how neurons communicate. But it turns out that work over the last many years is continually starting to implicate other cells in our brain called glial cells, the non-neuronal cells of the brain. And it turns out that cells like microglia, our resident immune cells, are part of this pruning process. They're actually coming in and associating with synapses during development, and work from our lab and others suggests that these cells are one of the reasons how these synapses are getting sculpted. Now, these are cells in our brains. These are our resident immune cells, and interestingly, they're the only cell not born in our brains. They actually enter our brains very early in development, there's many different aspects that make these cells particularly special. And one of them is that you cannot appreciate from this diagram is how incredibly dynamic these cells are. One of their jobs in the brain is that they're constantly surveying our brain environment. So if you can tell by this imaging movie, well, we can actually take a mouse that's been genetically engineered so that all of the microglia are labeled fluorescent green, and we can put a mouse under a microscope and watch their microglia. So right now, as you're sitting in your chairs listening to my talk, your microglia are doing this. And actually, that's a really good thing. They're sensing their local environment, and they're surveying the brain. They have a lot of homeostatic roles in the brain. And one of the things we wondered, especially in development, we've made observations that these cells were particularly active in developmental times and periods in which there was a lot of remodeling or pruning going on. And that raised the question, what are microglia surveying? And one of the things we discovered that they're surveying are the synapses, those connections between neurons. And so what I'm showing you here, if you could play the movie, is that this is a green cell, microglia, and what I'm showing you in this image is a neuron overlaid. And this is a branch of those neurons, and all those bumps are the synapses. And what you can hopefully appreciate from this movie is that the microglia are constantly touching those synapses. And when we did an experiment in a mouse brain where we had the ability to watch this in real time, what we discovered was that one of the things microglia are doing when they're touching synapses are literally eating or nibbling off these extra synapses during development, much like Pac-Man in our brains. Now, those that can appreciate, who knows what Pac-Man are? Okay, thank God. Okay, so Pac-Man, in this case, are the microglia. They're nibbling synapses, and actually, this is a good thing. This is not disease. This is what they're normally doing. This is their day job. And so this raised the question to us, and this is just an example of a microglia with lots of synapses inside their bellies during development. What we discovered is they don't keep doing this. They're doing this just during these critical periods in development, and then they stop. So now the question is, how do they know which synapse to prune and which one not to prune? So we actually can label the synapses in different colors, and in this case, they're red and blue. And here's our microglia right among a sea of synapses, and we asked, are they picky about which synapses they eat? And it turns out they are. They're very picky, which is something we'd want to have happen so they're not aberrantly just engulfing random synapses. It's a very precise process. And so this cartoon just simply illustrates a typical cartoon of a synaptic pruning process in which you have a neuron is innervated by lots of inputs, these red and blue ones, and then during development, some of them, the blue ones, normally get pruned, and then the red ones get strengthened and maintained. This is that use it or lose it idea. And what we discovered is that microglia preferentially engulf the blue ones. And that suggested that there must be cues or signals on those synapses that distinguish the red ones from the blue ones. What might be those cues? Well, this is the interesting part, because the cue we discovered was critical for this process is a group of immune molecules called complement. And this is interesting because what's the job of these immune molecules in the immune system is to tag a bacterial cell or a dying cell in your periphery for rapid removal, right? That's part of their innate immunity. And it turns out that a lot of these molecules were actually cues, and they're normally expressed in the brain, and that was how microglia were recognizing the synapses to prune. So for example, as I mentioned, in the immune system, these molecules called complement, I've noted just a couple of them here, C1q, C3, don't worry about the names. They're essentially coding or tagging that bacterial cell in your immune system, and the Pac-Man of your immune system, the macrophages, have receptors, and that enables them to eat or recognize bacteria. And what we discovered is in the brain, now this is a synapse. And the synapses are getting coded by complement, and microglia can recognize those synapses, much like the macrophages in the immune system. So here's an example of a mechanism used in the immune system that's being used in the brain to sculpt synapses, normally. So now the question is, okay, this is a normal process, it's tightly regulated during development, but what if this went awry? What if there was too much complement or too much microglia synapse eating? We think this could be underlying synaptic loss and impairment in disorders like schizophrenia and other diseases. And so this is sort of the emerging model from work now in my lab and now others, is that this normal pathway that I just described to you doesn't get turned off properly or it's overactive. And this raises the question of whether dysregulation of synaptic pruning through a mechanism like this could contribute to the pathobiology of some of these diseases. And indeed, work that I'm going to tell you about briefly today are implicating this pathway in schizophrenia as well as Alzheimer's disease. Now, this is going back to this classic image or figure from David Lewis's lab that suggests that in schizophrenia, by looking in postmortem brain in the frontal cortex and looking and counting all those synapses, that there's a decrease or a sparsity in these connections. And what I'm pointing to, these bumps or spines or synapses that I showed you earlier. So the question with this is, when did this emerge? Is this cause or consequence? So this is often looked at years after someone develops schizophrenia because it's post-mortem brain. So the question is this cause a cause, and what are the mechanisms underlying this? And this is where emerging genetics has implicated the complement cascade. And this is a really pioneering work by Steve McCarroll and Ashwin Sekar and many others have actually pointed to the very pathway I just told you about that we've been studying in the developing mouse brain. And this led to a really exciting interdisciplinary collaboration between my lab and his lab and many others at the Broad and Stanley Center. And essentially, this goes back to the discovery from the genetics, which shows that there are many genes that are now being linked to, or loci that are being linked to, increasing risks of schizophrenia. And this is just a Manhattan plot identifying these peaks. And the tallest peak of this Manhattan plot is at the MHC locus in the human genome, which contains hundreds of genes, and it's been a big mystery which gene actually is responsible for this genetic risk. And work by Steve and Ashwin actually honed in on that loci and identified C4, which is a gene right in that classical complement cascade that I was just telling you about, as contributing to a lot of that risk. And it turns out what they discovered, very quite simply here, is that those individuals that have a certain form of C4 make too much complement, make too much C4 in the brain, and this is associated with more risk of schizophrenia. And so this is now a question of how this relates to pruning. And when we looked in the brain, both human and animal models, C4 was decorating synapses in the brain. And work now by many of us together are suggesting the possibility or the hypothesis that too much of this tagging could lead to too much synapse loss. And of course now, collectively, it's suggesting a model that we're actively testing in the lab, which is that over-pruning through this or other mechanism could lead to connectivity defects because of the synaptic loss, and whether or not this could ultimately relate to some of the impairment observed in these disorders. And if we could understand the mechanism, what turns this on, what turns it off, and understand the biology more deeply, we're hoping this could point to new clues and new mechanisms that could someday give us a therapeutic window as well as potentially new biomarkers as well. So we are also now looking at the other genetics and starting to think about whether other genes could be converging on this pathway. And so there's much work left ahead, but the genetics in this case has converged with biology in a way that we're in a position to start to model this pathway in many different ways, and also to try to validate this disease mechanism in humans. Now, I started off by telling you that synaptic loss and cognitive impairment is a hallmark of many brain disorders. And I just told you about neuropsychiatric disorders like schizophrenia, but it's also a hallmark of aging brain, neurodegenerative diseases, including Alzheimer's disease. In fact, synapse loss is happening in Alzheimer's disease years before the first signs of cognitive impairment. And that raises an intriguing question. Could there be common mechanisms or converging mechanisms that are actually linking all these diseases together, although they're so different with respect to the timing, the age of onset, and some of the symptoms? Could this be a common denominator? And could we learn from one disease and have that try to help us to understand another? Well, it turns out that, as I mentioned, in Alzheimer's, we decided to go after this question as well because it had been well studied both in human patients, and there's actually some animal models that also showed this early synapse loss. And even in the preclinical stage of Alzheimer's, synapse loss is one of the earliest events. So again, what makes synapses vulnerable in Alzheimer's? It's the same question I just asked for schizophrenia. Could we understand the mechanisms and especially this early synapse loss is important. Could we understand how this happens so that we can try to come into an earlier therapeutic intervention? Well, it turns out, and this is why I'm bringing us back to the microglia, the genetics, once again, are pointing a laser literally right on these cells, my favorite cell, microglia. So most of Alzheimer's disease is late onset. In fact, almost half of the genes now implicated in late onset Alzheimer's disease are either expressed exclusively in or enriched in those cells, microglia, or they're related to the immune system, including the complement cascade. So this might be a coincidence, but it's made us more than ever want to understand the biology of these cells. Before we can understand what the genetics are telling us, and this is just not to worry about the names of the genes, but just to show that in these common variants, right, there are many of these blue dots that are implicating immune system in the microglia. So we need to better understand the biology of these genes and how it affects microglia function. And in a recent study by my lab, we actually went and looked in Alzheimer's mouse models, and we asked whether the same pruning pathway I just told you about with complement and microglia could be leading to early synapse loss in Alzheimer's disease. And since there were some animal models we could test this hypothesis in, we showed that indeed complement proteins, and this is in green here, were similarly tagging too many synapses in vulnerable brain regions like the hippocampus and the frontal cortex. And when that happened, we got too much synapse loss. And excitingly, at least in a mouse model, when we blocked this pathway, genetically or with therapeutic blocking antibodies where we could actually block some of these components, we could protect some of the synapse loss in some of the cognitive impairments, at least in a mouse. So now the challenge lays ahead of whether we can use this information to try to validate this and sort of translate this into human disease. Now, microglia, as I just mentioned, we focused on pruning today, but they do so many other things than prune. We're just scratching the surface of trying to understand their biology. And another common hallmark of many brain disorders, including Alzheimer's, but it could also be brain injury and other disorders as well, is that these cells, microglia, that I just told you in that movie, were dynamically surveying your brain. They undergo dramatic changes when there's even a local insult or injury or challenge. And this is termed microglia activation. It's a word we don't really understand, but we know they change. And the challenge there is because they have so many roles in the brain, when you see a microglia change shape, what is this actually telling you about what they're doing and how they might be going awry? And so the challenge there is because they play so many roles during development, they undergo so many different states, ranging from pruning to helping to secrete growth factors and many other things they do in the brain. When there's an injury, they shift to these other states. They can promote inflammation in the brain, which would be a bad thing, but there's also good inflammation that helps protect the brain. I just told you about pruning. That would be considered a bad thing, having too many synapses get pruned away, but their ability to eat things, to be Pac-Man, can also be really helpful because they can actually clear debris or a toxic protein like beta amyloid. So because they have all these roles, it's very clear that we can't just blanket target them. If we block all microglia, we're going to block all their good functions. So it's critically important that we start to understand these different states and that we start to find ways to target their specific functions at the right time. And I think if we could do this, it's going to open up new possibilities in thinking about microglia as a therapeutic target and as a biomarker. And we're particularly interested in Alzheimer's as a first step because the genetics are telling us they're important. But I believe that if we can understand this, it could have broad implications in many disorders and in general for brain health. So understanding how these microglial cells change state and then mapping those state changes, identifying specific populations of microglia relevant to specific functions, that we think might then allow us to develop more specific targeted approaches to switch them from the bad state to the good state, if that makes sense. So now there's efforts underway using new technologies called single-cell sequencing, where we can profile individual microglial cells to try to better understand why and how different populations can have these different roles in disease. And I think by doing this, not just in animal models, but in the human brain, we actually can do this now. This is going to identify new disease mechanisms, and I think even early biomarkers, we can imagine some of this might yield to new PET ligands and imaging biomarkers that can enable one to track these cells over time. And they may then also provide insight into when synapses are vulnerable. Imagine if we could go into our brains and know when our synapses were starting to become dysfunctional, early, before any symptoms. That would be really amazing, and I think we're on the road to be able to start to do those types of things. So thanks so much. I just want to end by thanking an amazing group, my lab in particular, and so many amazing collaborators. It's been so critical collaboration for all of this work and other work in the field to move forward, and also generous funding and support. So thanks so much.